Hi everyone, I'm going to discuss our experiment on demonstrating contextual control using visual stimuli in zebrafish. Before we dive into the exciting world of zebrafish, it might be helpful to review some of the basic keywords relevant to this experimental study. If we recall Skinner, operant conditioning is when consequences that follow a behavior affect the frequency of the probability that that behavior will occur in the future. When a contextual factor is correlated with the operant over time, they begin to come under some level of control too. An SD is a contextual factor that increases the probability of that behavior being emitted in the future. On the other hand, stimulus delta is a contextual factor that decreases the probability of that behavior in the future. Lastly, differential reinforcement is quite simple. Provide reinforcement for desired behavior, and don't provide reinforcement for undesired behavior. Although we enjoy the concise simplicity of the three-term contingency, behavior is not really as easy as 1, 2, 3. When a behavior comes under operant control in a specific context, the discriminative stimulus emits a specific behavior. However, the role of the discriminative stimulus may be influenced by presence of another contextual condition. Contextual conditions can include a variety of variables, including color, light, smells, temperature, hunger, pain, etc. As research within applied behavior analysis continues to expand, studies demonstrating relationships between behaviors and environmental conditions are far more complex than previously thought. Consequently, more research is required to examine how contextual conditions influence discriminative stimuli across animal research. The worldview of contextualism explains that behavior needs to be analyzed in context, because that context provides meaning to that behavior. Adopting a CVS approach allows researchers a deeper analysis into the function of behavior. Zebrafish have been used in a variety of studies examining the structural and chemical effects of various drugs and toxins. Beyond manipulating the fish to examine genetic changes, zebrafish have also been used to demonstrate visual discrimination, spatial discrimination, and a variety of behavioral tasks. They have demonstrated the ability to discriminate across several different conditions in a variety of different ways. The purpose of our study is to expand on the literature by demonstrating discrimination across contextual conditions. We use differential reinforcement across both conditions. Later, we will examine our results using a CVS approach. The subject of this study was one adult zebrafish. The fish lived in a freshwater habitat and ate generic flaked fish food from a pet store. The subject was exposed to 12 hours of light, followed by 12 hours of darkness each day. The experiment was conducted in a small tank over the course of 2 consecutive days. During the experiment, a white wire was placed across the top of the tank to divide the tank into 2 equally sized sides. The left side of the tank was designated as Condition 1. During Condition 1 procedures, a blue piece of paper was used as a visual contextual stimulus. The right side of the tank was designated as Condition 2. During Condition 2 procedures, a white piece of paper was used as a visual contextual stimulus. An AB-AB design was used for the purposes of this study. The independent variable was the application of tapping on a fixed interval schedule. Every half second that the subject was emitting the target behavior for that condition, tapping was applied to the side of the tank. The dependent variable was the duration of seconds that the subject remained on the designated side of the apparatus. A trial began when the contextual stimulus was placed on the tank for the conditions being observed. Trials were alternated between conditions, and each trial was conducted 30 seconds after the termination of the previous trial. Time recording began as soon as the subject entered into the designated area of the apparatus for the specific procedural condition. Duration was recorded in seconds from the time of the fish entered the designated area to the time the fish exited the designated area. Each trial was terminated upon the exit of the designated side of the tank. Trials were also terminated in the event that the fish did not swim to the designated area within 10 seconds of its start trial. Differential reinforcement was not provided during baseline procedures. Baseline procedures were conducted until steady responding was achieved. During intervention trials, procedures were similar to the baseline procedure, except differential reinforcement was provided contingent upon the target behavior for that condition. During condition 1, when the fish entered the left side of the tank, tapping was provided to the tank. Tapping was provided for every half second that the subject remained in the designated side of the tank. When the fish crossed over to the right side of the tank, no tapping was provided, and the contextual stimulus was removed. During condition 2, the same procedure was conducted, except the target behavior was to remain on the right side of the tank. Intervention trials were conducted until a subject met the criterion of a 20-second duration for 3 consecutive trials for each condition.
Are we cutting it off at 20?
What is it at right now?
It's at 37, but it didn't go in.
You see it, okay?
Our results indicate experimental control across both contextual conditions. Baseline rates were significantly lower than that of the treatment phases in both conditions. When we compare performance, we find varying patterns of responding across both contextual environments in the presence of differential reinforcement. Data from both conditions indicate that when differential reinforcement is withdrawn, the duration of the target behavior drops back down to levels similar to those in baseline conditions. Our results support the literature that fish can learn through differential reinforcement across contextual conditions. For us, this was a blue piece of paper on the tank wall and a white piece of paper on the tank wall. Upon analyzing our results and methodology, it was interesting to see how our behavior was shaped based on the behavior from the subject. The CBS approach encourages researchers to apply the same behavioral principles to ourselves. Doing so allows us to accurately communicate the context to which our results apply. Although significant efforts were made to rule out any threats to internal validity throughout the study, the fish in the study was assigned and not randomly selected. Additionally, current research indicates that tapping may initially act as a reinforcer. However, over time the fish may become satiated and the tapping can adapt itself as a neutral stimulus. Another limitation of this study is that it was conducted over a short amount of time. Research could be further expanded by examining the effects of contextual conditions, such as tank environment compared to that more of a natural environment, or how rapidly fish can demonstrate operant control. Other areas of expansion could include examining various topographies of contextual conditions, examining contextual control across other animals, and examining how to overcome reinforcement satiation across contextual conditions.